Hello, this is Dr. Bates, and we're talking about Chapter 16. Chapter 16 is going to go over innate immunity, the non-specific defenses of the host. So first, just a question for you to think about. During 1 year, nosocomial infections occurred in 74 patients in the hospital. All of those patients were incubated and mechanically ventilated. The infections were caused by Burkholderia, transmitted in non-sterile mouthwash. Now, these weren't the only patients who used the mouthwash. Other patients used it and did not have any problems. Why was it these patients that actually had an infection and not the others? Okay, so we're going to talk about the immunity. We're going to first bring up innate and adaptive, and then go into innate for this lecture, and adaptive is the next chapter. We're going to talk about the first line of defense, the second line of defense, antimicrobial substances, including complement, mention interferons, iron-binding proteins, etc. So first, some terms. If we talk about immunity, we're talking about people who are resistant versus people who are susceptible. Now, I've used those words before when I talked about the disease triangle. So, susceptibility is the lack of resistance to a disease. Immunity, or resistance, is the ability to ward off disease. If you have innate immunity, it is defenses against any pathogen, whereas adaptive is to a specific pathogen, and we're going to define that as we go through this. So first, innate immunity consists of our first and second line of defense. Our first line of defense is our skin, mucous membranes and their secretions, and your normal microbiota, okay, the things that are on the outside per se. Your second line is where the organisms have gotten past your first line. The second line is more internal. We have our phagocytes. We have our inflammation process that helps get what you need to the site of infection. We have fever, and we have antimicrobial substances. And then our adaptive immunity is our third line of defense, and that's where we talk about T cells and B cells and antibodies. And this is specific. The other one is to anybody on the outside, anybody who's made it past the skin, and these are going to be general, any pathogen. Okay, so first the big picture, just to put this again, we already went through that, but outside of your body you have possible pathogens, which include viruses, bacterial, fungi, worms. Your skin, mucous membranes, and antimicrobial substances is your first line of defense. It's on the outside. They get past that, then you have your inflammation, fever, and your phagocytes. And then your last line of defense, or the most specific, is the humoral and cellular immunity. Now here, in this one, we see our cells. So we're going to break it innate and adaptive. Of course, this could be more in depth. If you take another class, you could take a class just on this. But we are going to be general because we are talking about microbe instead of A and P or anything else. Your white blood counts measure the leukocytes in your blood. If you have high white blood counts, then you could have a bacterial infection, an autoimmune disease, or some kind of side effect from medications. If you have low white blood counts, that could be viral, pneumonia, again, an autoimmune disease, or a cancer. So here's just a kind of a picture showing how much, if we just had a volume. So we are mostly plasma and red blood cells, and then that small in the middle are our white blood cells. We have a surveillance system in our body, and our surveillance system actually can look for bad guys. So host cells have toll-like receptors that are attached to pathogen-associated molecular patterns. When the toll-like receptors see the bad guys, that's what the pathogen-associated molecular patterns. So basically, every cell in your body has a name tag, and every invader has some kind of molecule on the outside of its cell that makes it identifiable. And all cells of that type tend to have the same molecule. So your toll-like receptors are actually looking for things that don't belong. Non-self molecules on things that don't belong. When the toll-like receptors bind to something, when they recognize a possible invader, then they're going to induce cytokines that are going to cause or influence immune responses. Okay, so first, physical factors. Our outer layer is our skin, and our skin is a physical barrier to stop microbes from getting in. So if you have dry skin, if you have scratches on your skin, if you have breaks in your skin, then your immune system is compromised. We have the dermis and the epidermis. The dermis is actually the inner portion. And the way this works is new cells start here, and as they get older, they get pushed to the outside. Shedding and this environment that has a lack of water actually makes your skin inhospitable to most microbes unless they figured out how to get past that lack of water. Your mucous membranes are in your gastrointestinal, respiratory, and genitourinary tracts. And these mucous membranes produce glycoproteins that get microbes stuck. If we talk about, let's say you have drainage, sinus drainage, that is there to actually wash out that area and carry those microbes down into your digestive tract where they're going to die. It also helps prevent those tissues from drying out because of where they're at. Now, an example of something that happens in your lungs is a ciliary escalator. This is where microbes that get trapped in the mucus actually get waved away, if you will, where they're actually going to be pushed out of the system. That mucus is going to be cleaned out. That's going to keep microbes from being able to infect the cells there because the mucus is protecting it. We also have, of course, the way our eyes are set up, because that's another way for microbes to get in. We are going to have saliva in our mouth that's going to wash microbes out, and not only a physical factor of the liquid moving, again, down in your digestive tract where things are going to get destroyed, but we also have some antimicrobial substances in that. We'll mention that when we move to the chemical, but there are some there. Urine, the reason you pee a stream is to actually clean out the system. That is going to blow it out, if you will. Vaginal secretions are also going to be a flow out, basically taking things out of the system. Chemical factors. We have quite a few. We have sebum, which is a fungistatic fatty acid. It provides a protective film, and it keeps pH low. If you remember, back when we talked about microbes, we talked about what pH would have an effect on them. So your body maintains a pH. Lysozyme is a chemical that you're going to find in your saliva. I mentioned that we have chemicals, your tears, urine, all those things. They're going to actually destroy bacterial cell walls. Your gastric juices have a low pH. That's going to destroy, hopefully, most of those bad guys and toxins. And then vaginal secretions also have a low pH, which is going to inhibit microbes. We inhibit microbes, but we also want microbes. We want particular ones. We found that microbes play a big role in our immune system, and the more we find out about them, the more we find that they play a role in our health. So microbial antagonism or competitive exclusion, I call it a crowded beach. These are where your normal microbiota, the ones you have, compete with pathogens, or they alter the environment. Now, some people are actually arguing that microbes are actually part of your genetics. Now, that seems kind of funny because they're not part of your DNA, but they are given to you by your family. Your mother and father play a role in making your first crowded beach. So does every other person that baby touches. So you might want to think twice before you let your stinky Uncle Bob play with little Bobby as soon as he's born. A commensal is a normal microbiota. The catch is that the word commensal means one organism benefits and the other is unharmed. But we actually have a symbiosis because microbes who grow in your skin help you by doing this. Okay? Now, you could argue that if we're talking about opportunistic pathogens, they're somewhere in between. Until they actually infect you, they don't harm you, and they don't necessarily benefit you, I guess, but they're still there. So you'll hear the word commensals referred to for microbiota because that's the history of it. Now, of course, we have probiotics that you can take. Those are live cultures that you're hoping will create a new or increase the good guys in your system. Now, when we talk about cells in your blood, We have particular cells that do particular jobs. Now, this is microbiology, so the cells themselves, we're going to talk about their jobs, we're going to highlight some of them. You are not going to have to know the same detail you would in another class because we are talking about microbes. So we have our erythrocytes, which are red blood cells. We have our leukocytes, which are white blood cells. And then we also have platelets. These are created in red bone marrow stem cells. Stem cells can become any of these cells. They are going to differentiate into the main cell and then differentiate into the particular cell that does a job. Now we have granulocytes, which are named literally because if I look at them in a microscope, I can see granules in their cytoplasm. Those are the neutrophils, the basophils, and eosinophils. Neutrophils are phagocytotic, and they're important in the early stages of infection. Basophils release histamine, and they work in allergic responses. The eosinophils, again, are phagocytotic, and they're important for parasites and helminths. Now, the agranulocytes are the ones that don't have visual granules when I look in a microscope. They include the monocytes, and I'm normally going to call them macrophages because those are the ones I've actually dealt with. These guys are phagocytotic. The dendritic cells, you find those in skin, mucous membranes, the thymus, and again, they're phagocytotic. Then we have our lymphocytes, our T cells, our B cells, and our natural killer cells. Those are important for adaptive immunity. Natural killer cells, I kind of place in the middle between adaptive and innate, and we'll talk about that a little later. So here's just kind of putting them all out. We have our red blood cells and their job. Then we have our granulocytes and our agranulocytes, and then our platelets. The platelets are there, of course, for blood clotting. The guys in the middle are the ones that their job is to kill or initiate processes that are going to destroy invaders. In the lymphatic system, we are actually looking at a process where we are basically surveilling the system and asking, are there any bad guys? Literally, we're going to collect things. We're going to put this through the system, and we're going to ask, is there something that's not supposed to be here? So phagocytotic cells are cells that eat, engulf other particles. They will bring those particles in. In this case, we have a macrophage. It is going to grab a hold of the bacterium and actually bring it into the cell and destroy it. Now, example of phagocytotic cells are neutrophils, fixed macrophages, which means they're not moving around, and they're in particular tissues, and there are wandering macrophages, which actually move through the system. Here is the process of phagocytosis. The cell, let's call it a macrophage, is going to form chemotaxis. It's going to go somewhere. If there's been a signal that says there's a bad guy here, it's going to be attracted to that chemical signal and go there. Otherwise, it'll survey where it's at. It's then going to grab a hold of the microbe or the foreign particle. It's going to bring it into the system or ingest it by putting it into a phagosome. The phagosome here is going to be a container in which that foreign object is held in. It's going to then add the contents of a lysosome to that structure. That's going to allow it to basically digest that microbe. It's going to digest it into pieces. Anything that it can't use, it's going to kick out of the system. There's a couple things in addition to this, and I'm going to mention it in the next chapter actually, and that's how this cell plays a role in bringing up of future signals that, hey, I found a bad guy. I'll mention that again. So again, we have chemotaxis. That's going to attract phagocytotic cells. We have the adherence, where it actually grabs a hold of the phagocyte. We have the ingestion of that foreign object. Opsonization is something I'm going to mention a little later, and that's where a microbe is actually targeted. So the microbe is coated with proteins that makes it easier for the macrophage to grab a hold of it. Finally, that foreign invader is going to be digested into small pieces so it no longer can cause harm. Cells also can do something called an oxidative burst. This is where we actually release hydrogen peroxide that will kill the bacterium. Now, we mentioned how microbes can get past this, which is why I wanted to bring it up here. So we mentioned the pentose phosphate pathway. That actually allows the cell to produce superoxide dismutase, have hydrogen peroxide, and uses that to kill the bacterium.